Hello, and welcome to this explainer. Today, we're putting on our detective hats to explore sequencing scale and clinical best practices. Think of the human genome as our species' ultimate, incredibly complex instruction manual. It literally dictates everything, right, from your eye color to your risk for certain diseases. Today, we're exploring exactly how modern sequencing approaches let us read this massive manual to hunt down the specific variants that cause genetic conditions. We're going to look at the tools we use, the absolute staggering scale of the data, and the clinical strategies that guide us.
Now, I want you to really think about this for a second. How do you find a single typo in a 3 billion letter manual? Because when a patient presents with a genetic condition, that is quite literally the challenge we're facing. Just one single altered letter in our genetic code can dramatically impact a person's health. But randomly searching through 3 billion data points? Yeah, that's impossible. So to solve this, science has developed different skills of sequencing. Our journey today is going to start micro, looking at the tiniest, most precise fractions of our DNA, and then we're going to zoom out exponentially.
Okay, let's dive into this by zooming in on targeted testing. This is where we start our detective work, using the most focused tools in our toolkit. You see, you don't always need to read the entire manual if you already have a strong clinical suspicion about where the typo actually is. Targeted testing is used when clinical symptoms strongly point the finger at a specific genetic culprit. These approaches operate on a highly precise scale. First up, we have variant-specific testing. This is microscopic. We are checking roughly one single base pair, literally one letter, for a known mutation, like what we see with sickle cell anemia. But what if we know the suspect gene, just not the exact spot of the mutation? Well, that's where gene-specific testing comes in. We scale up just a bit to sequence a single gene linked to a disorder, like phenylketonuria. It's basically the equivalent of proofreading a single page instead of just one word. Sometimes, though, the symptoms could be caused by multiple suspects. In that case, we scale up to what we call targeted gene panels, analyzing roughly 1 megabase of data. Here, we check a curated group of genes associated with specific symptoms, like running a hereditary cancer or cardiac disease panel. We're investigating a cluster of known suspects all at the exact same time, which makes our search incredibly efficient while still keeping things highly targeted. And this brilliantly illustrates the main dividing line in genetic medicine. Targeted approaches are fantastic, but they completely rely on having known suspects based on clear clinical presentations. But what happens when the diagnosis is a total mystery? When the symptoms just don't point to one specific gene or even a known panel of genes? Well, that is when we have to abandon our targeted search and cast a much, much wider net. Which brings us to our next approach, going broad with exploratory sequencing. When we don't have a clear suspect, it's time to look at the bigger picture. When targeted methods don't yield answers, or when the clinical presentation is entirely unclear, we transition away from proofreading a single page to scanning huge volumes of the genetic text. The data explodes exponentially. Our first major exploratory tool here is whole exome sequencing. At roughly 45 megabases of data, this is a massive leap. But here is the absolutely critical part to remember. Whole exome sequencing isolates and sequences only the protein-coding regions of the DNA. We are looking strictly at the parts of the manual that actually build and run our bodies, mainly because that's where a large majority of known disease-causing mutations are found.
Now, what's really fascinating is just how specialized that exome actually is. The whole exome, all those crucial protein-coding regions we just talked about, makes up roughly 1.5% of the total genome, just 1.5%. By sequencing the exome, we temporarily ignore 98.5% of the genetic code to focus exactly where the most vital clues usually hide. When you stop and think about it, the vastness of that remaining, unread DNA is truly staggering. Let's move to and see how this builds into our largest scale yet. Sometimes, even the whole exome isn't quite enough. When we absolutely must read the entire book, cover to cover, we turn to whole genome sequencing. We have now reached an enormous 3 gigabases of data. We're reading the entire sequence, including that massive 98.5% of non-coding regions, leaving literally no genetic stone unturned. It is the ultimate exploratory tool.
But as you can probably guess, accessing the entire genome comes with immense weight. Whole genome sequencing requires the absolute highest amount of sequencing volume. You aren't just generating the data, you have to store it, map it, and interpret it. Finding a novel, completely unknown variant in 3 billion base pairs takes incredible computational power and highly sophisticated bioinformatic tools. It is a monumental undertaking compared to just checking a single base pair. So we have all these incredibly powerful tools at our disposal, but the science is really only as good as the strategy behind its application. Let's shift gears a bit and look at best practices in testing. Genetic testing isn't just about a single individual. It deeply impacts entire families. And navigating that requires a highly structured, empathetic clinical approach. This brings us to what we might call the clinical golden rule in genetic diagnostics. When you have a family seeking answers, it's like a complex logic puzzle. You have to follow a very specific strategy. Step 1 is always this. You absolutely must identify the causative variant in the presenting affected individual first. You use your exploratory or targeted tools to pinpoint the exact typo causing their symptoms. Once you have successfully identified that specific variant, then and only then do you move to step 2, which is running targeted tests on family members. Notice the strategy here? You aren't running whole genome sequencing on the rest of the family. Because you already solved the mystery in the affected patient, you can now simply run a variant-specific test on the relatives to check for the presence or absence of that exact same variant. So the crucial point is this. Success in clinical genetics is entirely about matching the right scale of sequencing to the clinical reality right in front of you. You don't sequence an entire genome if a one-base targeted test will do the job, and you certainly don't screen healthy relatives with broad exploratory methods when you can first identify the specific genetic culprit in an affected family member. By understanding scale and by sticking to best clinical practices, we can effectively navigate those 3 billion letters and solve some truly complex family genetic mysteries. As we wrap up this explainer, I want to leave you looking back at the sheer scale of what we are working with. We know so much about that 1.5% protein coding exome, but as whole genome sequencing becomes more and more accessible, it begs a really fascinating question. What secrets are hiding in the other 98.5% of your genetic code? The non-coding regions are vast, they're complex, and they are still largely uncharted territory. Our instruction manual definitely has plenty of mysteries left to solve. Thank you so much for joining me today. Keep asking questions and keep exploring.